Hello and welcome everyone to our free webinar on bioinformatics and data science. Today we'll talk about upcoming programs that are designed for beginners as well as high school students, and we're going to continue to explore bioinformatics in the context of different types of omics data. So briefly speaking, we will have two upcoming programs. These programs are going to be focused on bioinformatics as data science with applications to biomedical data sets. We will have a program designed for data science and machine learning focused on statistics and methods. And we will have a program that explains the relationship between structural biology and sequencing data. So these two programs are enriched with materials that you probably already know that our company has been offering through a variety of different portals. We're working on preparing additional materials, including coursework, as well as technology that allows us to track and interact with you as you go through the various aspects of these topics that we will cover. We take and use some of the publicly available data and published methods in top research journals, and we offer online certification that you can share and use to build up your resume and develop skills that are needed in the workforce. So in this webinar, I will briefly cover many of these topics that are focused on two specific programs that are coming up. But in general, I will also discuss some of the aspects of these materials that we have prepared for this program, for these upcoming programs specifically. Briefly about myself, my name is Ilya Brodsky. I am the CEO of Pine Biotech, and our company is on a mission to help researchers to harness their data and understand how to apply high statistical and machine learning methods to high throughput omics data. Our company was co-founded by Dr. Alfred Tauber and Dr. Leonid Brodsky, but we also have a number of other team members that are going to be closely working with us to make this program possible, including Julia Panov, Avi Titievsky, Mohit Mazumdar, Joshua McKendall, and Bipsa Biswas. We have quite a lot of experience working with a variety of students in different settings. Primarily, our background is at the college and university levels, and we interact with a lot of researchers that apply the tools and the learning programs that we have to understand and apply these tools to different types of omics data. So what is omics data? Omics data is really a collection of different types of data, including phenomics, genomics, epigenomics, transcriptomics, metagenomics, so many different types of omics. And each one of these different types of omics data is used now in all types of biomedical research and all life sciences, really. So when we talk about these applications, they are important for all steps of data generation, processing, and interpretation. So that's why these programs are designed for anyone that interacts or plans to interact with this data in the future. For example, those in the lab generating and they need to generating data who need to understand how to generate more and better data about what's going on. Those that understand the data and seek to analyze that data and integrate it with other types of data, and those that need to interpret that data to make decisions about experimental design or even about the application in the clinic or in the biotechnology and pharmaceutical sectors. So that's why we believe that it's important to understand biology that is becoming more and more a data science. And our company is focused on developing tools and resources to enrich bioinformatics training and offer it at the earliest possible level and support the growth of bioinformatics all throughout the professional development. So that's why we are introducing these two new programs starting in July. One is going to be called Omics 2020, and another one will be called Genomics in the Virtual Lab. So these are two different programs. They do overlap, So for those of you that are thinking to join one of these programs, you can either choose one of them or both of them. If you do choose both of them, please take a note of the timing. So Onyx 2020 will start on July 6th and go all throughout July 17th. The timing will be from 2 to 4 p.m. Eastern Standard Time. And the Genomics in the Virtual Lab will be July 13th to 24th, and that will be 10 to 12 a.m. Eastern Standard Time. So for those of you that are joining us from international locations or from other states with other timing, it's important to take note of that as well. Okay, one second. Okay. So also please note that we have a chat box here that you can see. And we also have a stream on YouTube. So you will be able to ask questions. And we have here Beepsa and Mohit that are interacting with you. So they will be answering any questions that you might have. And also we'll be able to share the specific links if you are interested in viewing some of these program details. Okay, so these programs are going to be led by highly experienced bioinformaticians and biostatisticians that are on our team. You will see them either directly guiding some of the elements of the program or supporting by providing directions and tools as well as resources. And primarily they will include Darko Medin, who is a biostatistician and a data scientist, Julia Panov, who has a PhD in bioinformatics from Haifa University, and Mohit Mazumdar, who also has a PhD in computational biology and has ample experience in both research and working for pharmaceutical companies. We will also have guest lecturers, primarily in our program for genomics in the virtual lab. That will be partially handled by Dr. Sona Vasudevan, who is a professor and a program director of biochemistry at Georgetown University Medical Center in Washington, D.C. She has a lot of experience teaching both U.S. and international students. And so we're excited to have her participate in this program and share from her experience, sharing how she developed her skills in the omics data space, starting from working in for PDB and now teaching and doing research at Georgetown University Medical Center. So first of all, let me briefly introduce these programs and the topics that they will cover. The Data Science Omics 2020 program will be focused on the analysis of big data, bioinformatics, data wrangling, and machine learning. So these are specific types of skills that will require some elements of conceptual understanding of what these methods do and why they are used, and some practical and technical training, including some coding aspects of coding that will be available in this program. It will be a 2-week program. In the first week, we will talk about different types of omics data sets, challenges and opportunities related to big data. We will talk about how to clean, load, and process data, doing exploratory analysis, and then practicing some hands-on and specific elements of handling large and complex data sets. In the second week, we will talk about machine learning and artificial intelligence, where we will cover unsupervised and supervised methods for machine learning, talk about model accuracy and validation in order to really think about designing machine learning, processing, and analysis for the real situations that we will discuss in the context of real projects. The second program, Genomics in the Virtual Lab, is also going to be a 2-week program. And you can think of this program as an introductory program that will start from introduction to bioinformatics, discuss the relationship between protein structure and function, and focus on multiple sequence alignment and evolutionary analysis. including analysis of mutations and 3D structures of proteins. In this program, we will have guest lectures and different activities and games, and you will explore how different computational tools, including different types of 3D and 3D visualization and analysis software like PyMol and Chimera will be used. We'll also talk about different types of example datasets and projects like sickle cell anemia, cystic fibrosis, phenylketonuria, and COVID-19. So you'll also see how in this program, and I'll talk more in detail about all of these sessions independently, you'll learn how to access data, process that data, visualize that data, and try to interpret the relationship between sequence and function, looking at the biological impact of those sequence changes. So in the first week, we'll talk about bioinformatics tools and data sets, including visualization and finding the data. And after we go through this week, we'll have a weekend activity. During the weekend, you'll use some of the tools that we've discussed in the sessions to visualize specific proteins that are important for a condition that you might want to select. And we will do this so that you can really apply the skills that you've learned about to a data set of your own. And then in the second week, we will kind of focus on the big data and the availability of different types of data sets that require both processing and interpretation. So in the second week, we will talk about literature review and how to design your project and how to prepare your data set, how to find it, how to analyze it, and how to interpret it. And the outcome of this second week is going to be to really develop a project and present it in front of guest judges that will provide you with personal feedback on your project. So you'll end up, by completing this program, you will end up with a project presentation where you will learn how to analyze the data and explain the analysis methods that you've used, then present your analysis results, and get some specific feedback from experienced judges that come from both industry and research and academia. And we will also make sure that those projects are something that you can take with you. So in your certificate of program completion, if you have prepared and completed your project, that will be mentioned and you will get a certificate of excellence. So to register for this first program, Genomics in the Virtual Lab, we have a pre-registration process. During the pre-registration process, and if you can see in the chat here, the link, which is edu.tbioinfo.com, Genomics in the Virtual Lab. Let's take a look at this page really quickly. So as you can see, you will find a pre-registration page. It provides a lot of information on what will be covered in the program. You will also find the specific topics highlighted right here and some frequently asked questions explained down below. You will also find the program schedule, so you'll be able to see all of the different topics and what the sessions will include, including some of the practical activities and hands-on sessions. The registration fee is $250. But since right now we are anticipating a lot of interest outside of the US, please do reach out to us about registration fees and the application process. By starting, you can start by filling out this information right here, and we will be reviewing this information to determine eligibility for the program and making sure that you have appropriate background to join and benefit, and you can also fit it into your time. All of the sessions are going to be recorded, so you don't have to be necessarily in all of the theoretical lectures, but we do encourage you to participate in most of the practical sessions that you will see. So after you submit this form, you will receive some additional information, and we will be reaching out to you to make sure that you understand the process registration. And after registering and completing the registration process, you will have access to this organizational page, which is designed to support you with a forum where discussion will take place, additional resources like courses and projects that you can have access to, and tracking of your progress. And I'll talk about this a little bit more in the later part of this presentation. You'll also be able to interact with some of the other participants of the program, and you will be able to learn about them and potentially even collaborate with them on a project of your own. Okay, so this is the program called Genomics in the Virtual Lab. Did anyone have any specific questions about this program that I can answer right now? Okay, if you have any questions, please feel free to put those in the chat. Again, I'm specifically going to be focused on the chat here in Zoom. If you are watching this on YouTube Live, Vipsha, if you could please put it in the Zoom chat. Okay, so here's a question. How can I learn more about the topic of this webinar? Well, the webinar is really introducing these programs. If you would like to learn about the topics covered in this program, and I'm going to speak about the second program here in a second, you should go to this link, fill out the pre-registration form, and then we will be able to send you more information if you have some specific questions. Okay, what is the registration fee for Omics 2020? So I will speak about the Omics 2020 program here in a second. Are there any other questions about genomics in the virtual lab? Okay, here's another question. Are the current sequencing techniques developed enough for diagnosing rare hereditary diseases through embryo screening? Yes, I believe they are. And in fact, screening of those diseases happens, at least in the US. And a number of different panels have been developed. We will speak about the sequencing technologies and the processing of that data in the program a lot, and we will actually talk about some of those hereditary diseases. So if you're interested in that, you're welcome to join that program. Okay, what is the biggest difference between the two programs? So this one is primarily focused on the relationship between sequences and structures of proteins. And it is designed for high school and beginner level. And the topics and the examples are chosen appropriately. Let me cover some of the topics in the second program, Omics 2020, and then we will return to some of these questions to discuss the differences between the programs. So the second program is called Omics 2020, and it's really focused on analysis of big data and machine learning. So in this program, we will cover things like what types of biomedical data and data types and formats and challenges are either now happening or are coming up in the very near future. We'll talk about the exploration of these types of big data set to help us process and prepare it for analysis and understand conceptually where is the signal and where is the noise. We'll talk about machine learning, so how to apply machine learning to big data, and that data could be many different types of data, but primarily we'll focus on biomedical data and omics. And then we'll also talk about the interpretation. When we find insights in these big data sets, how do we actually apply it to what kinds of problems? So we'll talk about that. Now, this program is 2 week long. We have a number of different components to the program. The first week will focus on different data types, big data challenges, exploratory analysis, and really preparation for machine learning. And the second week, we will talk about machine learning from different methods and settings of those methods, and all the way to interpretation and production quality machine learning models. So what is machine learning? There are two different types of machine learning. The first one is when we ask the machine or the algorithm to detect patterns that could be discovered in the data, right? And that's called unsupervised machine learning. The second part is supervised machine learning, when we train a model to predict or classify a new data set. And so to understand how these different methods apply to different types of omics data, the first thing that we'll do is conceptually, we will utilize our Bioinfo platform, where a lot of these methods are introduced and you can apply them without any need for coding. In fact, in the first part of the program, we will look at how to process and then apply some of these statistical and machine learning methods to the data sets coming from gene expression, mutation variants, and metagenomics. So what is the Bioinfo platform? The Bioinfo platform allows us to package individual scripts in a interface, a user interface that is visual, and this visual interface helps you connect the dots using different methods organized into logical sequences called pipelines. These pipelines are enriched with color-coded maps, different methods and explanations of those methods, as well as a variety of visual outputs that you can get directly. What's important here is that this whole process is guided, and as a result, you understand the impact of reproducible workflows. But once you've understood the conceptual connection between the different methods and how they work on different data sets, we will actually take you to what's happening behind the scenes, and we'll actually learn about some of these methods in R and Python. So we'll learn about how the code works and how you can modify the code and change the settings of these algorithms to get outputs that are designed by you, that you are looking to present or interpret. So what are the applications? First of all, we'll talk about how to do data visualization, summary, and exploration, including understanding of what these different visualization methods do and how do they really apply to specific problems in big data sets. We'll also talk about different types of data mining and classification methods, including decision trees, hierarchical clustering, random forests, support vector machines, different types of k-means clustering and other types of clustering, linear discriminant analysis, and others. So in the process of this program, you will get familiar with all of these different methods and how they work, in what cases they are going to work, and in what cases they're not going to work. And then we have prepared a number of practical coding tutorials that you will be able to go through and complete in the context of this program. For example, how to do dimensionality reduction using principal component analysis, how to identify differentially expressed genes and understand what types of pathways are down or upregulated in gene expression data, how to do clustering and how to find organization of those clusters and understand how to interpret it from a biomedical or biological standpoint. And then in what cases do we actually use support vector machines, linear discriminant analysis, or random forest? What types of features do they provide us with that we can use for what kinds of problems? And so in the context of these practicing exercises, you will actually go through a structured process. This is going to be organized by both R code and Python code. So you can choose either one or try to do both. And we have a system that will guide you from understanding the problem to learning the code to modifying the outputs, modifying algorithm settings, and then applying it to a data set that we give you, or try it on a data set that you yourself want to apply it to. As you go through that process, you will have a series of tasks that you need to complete. And every task that you complete, you will be able to get some points for. So that is a process that will build up your profile and help you achieve the objectives of this data science program. We will talk about how these different methods are actually applied to biomedical research problems. For example, we'll look at diagnostics and cancer biology. We will talk about tissue and single cell sequencing. We will also talk about metagenomics and infection caused by different types of pathogens. Where do these projects and data sets come from? We actually take them from publications from top research journals that highlight important aspects of machine learning in application to real data sets and real problems that represent clinical problems. So when we talk about that type of clinical problem or a pharmaceutical or biotechnology problem, we actually will explain how the different types of data sets and different types of applications differ as you go from basic research to translational research to actual application in the clinic. And so we'll talk about these different projects in terms of application for clinical, like patient stratification, disease diagnostics, mechanism of disease onset and progression, and also pharmaceutical and biotech challenges like biomarker discovery or target discovery, drug repurposing, and repositioning. So we'll talk about a lot of these applications as we go through the different examples that we have prepared. The second program, Omics 2020, again, we have a pre-registration link. So you can see that in the chat, but let's go to this pre-registration page, and I'll show you what other information is available here. So this page right here will tell you some specific details, and you can read about all of these topics and see some of the topics that I am covering in this webinar, but you'll also see a little bit more detail here, including how to set up some of these tools on your computer and what exactly are you going to be able to learn in these different sessions that we have prepared. So again, once you have learned about these topics, you're interested to apply to this program, you can sign up. And once you sign up, we will look at that data. We will see where you're from and what is your level and what is your background. If you have any questions, we'll be able to answer those questions. And we'll also share with you any other resources if you have a specific interest or topic that you are pursuing. Okay, so any questions about this program first, and then I'll talk about the comparison between these two different programs. Okay, so I don't have a powerful PC, so how will I use the BioPython program? Okay, great question. Let me actually show you why we have designed some of these technologies around being able to process and analyze data on the cloud. So two specific technologies that I want to cover today. One, as I mentioned, all of these different methods are including our powerful T-Bioinfo multi-omics processing platform. Here you will see, I'll just show you a couple of examples, how essentially what your role is to prepare a workflow. You will be guided on what these specific methods do and how they connect together. You will see how your choice of methods will provide different types of outputs, and those different types of outputs could then be prepared for what's called downstream analysis. So here you can see me building an RNA-seq pipeline. The RNA-seq pipeline will generate a table of expression, and after I have that table of expression, I can do differential gene expression analysis. So once I click on End, you don't need any computational resources on your PC or your laptop to be able to look at the outputs, okay? The second example is actually the coding environment. This coding environment is, again, going to be guided. You can select whether you choose R or Python. So for example, here you see principal component analysis and you see all of the code in R. You will also get a console and you can run this code. This code runs in the browser, so it does utilize some of the resources on your laptop or on your computer. But you can see that what it provides us with is some standardized outputs And some of those outputs are going to also be incorporated into the grading process. So how do you actually take this from here onto your computer? You can just change this right here. You can modify the script. You can actually follow the process of learning the code, how to modify the visual outputs, how to modify the algorithm settings, and at the end, save whatever modified script you have to run on your own computer. What to do if you don't have a powerful computer? You can either try this and see how it works, right, and use it on smaller data sets, or we also provide the same capability that is going to happen without any kind of utilization of your own resources. So all of it happening in the cloud. And actually, that is one of the topics that we will discuss. What are some of these challenges with the actual big data sets, right? Because they do require a lot of computational power to process and really get it to a place where you can interpret it. Okay. Any other questions about the Onyx 2020 program? Okay, let's talk about the two different programs in terms of fees and outcomes. Okay, and then I will actually return to complete some of the things that I still didn't have a chance to finish. So the Onyx 2020, both of these programs are essentially 2-week programs. The 2 weeks are going to include some Q&A sessions. They're going to include some hands-on sessions. They're going to provide you with curated examples and a lot of additional materials that you can utilize. Both of them are essentially 15 days or 14 sessions essentially. But you do get a lot of other materials and longer account access to all of the materials even after the program is completed. So you will have it for 30 days. Onyx 2020 is $150. Genomics in the lab is $250. Both of these programs are led by experienced faculty, and you get individual certification for the courses that you complete. You will have a lot of guided material in these courses about different types of omics data and about different methods. And a lot of people actually utilize some of these to build up their portfolio and build up their profile. For example, here you can see somebody sharing some of these on LinkedIn and ultimately standing out from the crowd, right? So you will be able to complete the coursework, get a certificate of completion from the full program, and also work on an independent project. If you have that independent project, let me actually cover some of these examples and explain how these materials work. Okay, so as I mentioned, we have been working for a long time with researchers, academic centers, universities, and colleges to prepare a lot of these materials. This is the first time that we are offering these materials on a wide scale to people that even do not have a degree and are potentially in high school, but looking forward to getting involved in these kinds of topics. But we've prepared a collection of materials that include introductory materials on bioinformatics, genomics, transcriptomics, epigenetics, metagenomics, and machine learning. All of these materials are personalized, so you will be able to go through the different materials that we have and build up your profile. You build up your profile by completing specific assignments, doing the specific tasks that you're assigned, and ultimately we can track all of the progress and help answer and personally address any questions that you might have. At the end of the program, we also have a leaderboard. And on this leaderboard, we highlight those that achieve top positions on this leaderboard inside a specific program. Many of the elements of the programs are going to be designed around this platform. On this platform, as I showed you, you can interactively go through the process and understand what each method does without any coding background, without knowing a lot about the different methods, but really working through the projects to understand what these different things do. You will also see interactive visual outputs, and those outputs will help you understand what the methods do in the context of a specific project. For example, you'll be looking at the relationship between sequence and structure. And another side of this is for those of you that are interested in coding or potentially have some experience already in coding, we will have this environment where you will be able to go through the scripts, understand how the scripts work, understand how to modify the scripts, and then apply them to data sets of your own. To apply them to data sets of your own, we will actually guide you on how to read the literature to understand what types of data is used in a specific research project, to then understand what types of methods were applied in the discussed article, and then to combine this data and this method with your understanding of what else you have learned in the program, what other data sets you can find, and develop a novel and independent project. So we also will provide you with a lot of examples where you can start focusing on specific types of biomedical or research projects. For example, we have a series of projects that will be made available that include epigenomic analysis of asthma data, different viral and infectious diseases, changes in the transcriptome, different cancer-related projects, even agricultural and neuroscience projects. So ultimately, going through some of these examples, you'll be able to develop a project of your own. When you develop that project of your own, we have examples of college level students as well as even high school students. So here you can see, and maybe Beepse, you can share the link to the blog that talks about Hannah Davis, who did one of these programs and was able to complete a series of materials and develop a project of her own related to diabetes. So some of these projects, all of these projects are taken from publications. They are organized in a structured way, and then students have been reviewing those projects and using them to actually develop independent projects and get feedback from their peers and from experts. Now we are taking this program online, but you can still develop a research project that will eventually culminate in a poster presentation, which could be supplemented by a video presentation of your team reviewing the project analysis steps and the outcomes of that research study. So those are common features between the programs. And as I mentioned, the differences between those different different types of focuses. One is more focused on the relationship between sequence and structure and how to understand and work with those types of data sets. The second one is more focused on specific types of machine learning and data science methods with some element of coding. So the first program will not have coding. The second one will have coding. The first one will have some other tools like Chimera and PyMol, and we'll have different types of projects. In the second one, it is going to be more focused on the basic data types and specific types of methods in machine learning. Okay, so the question, will it help in chemotaxonomy and phylogenetics? The first program is more related to phylogenetic analysis and taxonomy. We'll talk about how to do multiple sequence alignment, how to understand relationships between protein sequences or nucleotide sequences, things like that. So that would be more the Genomics lab program. Okay, so I already went through this information, and let's see if you have any other questions that I can answer right now. Okay, if there are no other questions, I did want to highlight that some of the previous participants of our programs started off without any background, were able to apply some of these advanced methods in a very short time. That's one of the critical aspects of the program, is that actually the time it takes from learning about these methods to actually applying them to a data set of your own and doing that analysis will be, instead of years, it will actually take you just a few weeks. We are not promising that every single person will come out with an independent, completed, and analyzed project, but we do have repeat participants that have joined multiple types of programs and supplemented those programs with additional online resources that we provide on the platform, and help actually put those skills to use in the context of a narrowly focused program. We also have a number of people that come from biology background without any background in data science or coding, so people that just come from the lab and learn about bioinformatics for the first time. But we also have a number of participants that have previously joined that are postdoctoral and faculty level, that are able to benefit from some of the materials and some of the examples that we cover in these programs. I do want to mention that even for those of you that potentially have faculty and teaching experience, some of the participants that we've had have found these courses informative as well. And we really try to incorporate a lot of the aspects of the feedback throughout the program to improve the materials as we go along. And we realize that the programs are short. They do not provide sometimes enough depth and enough time to simply complete a project during that specific program. So that's why first we allow you to have longer time with access to tools, resources, data sets, and even getting feedback from the program organizers and trainers. But we also offer a specific program that is focused on research internship and research fellowship. So that is another opportunity that I just wanted to highlight for this session. These programs are training programs. The training programs will walk you through a specific syllabus or a curriculum. As a result of going through that curriculum, you will acquire specific skills. You probably will develop a project idea, find the data set, and understand how to analyze it. If you don't have enough time, typically a project that requires that you want to take all the way to publication level, or a poster, a serious poster at some kind of a research conference, that might take between 3 and 6 months. So we've designed this research internship to specifically work for those that join some of these training programs and then would like to continue to work on their independent project, applying the cloud-based tools that we have, getting support and regular sessions where they interact with others, and have access to all of the different program sessions, right? So if you are thinking, oh, okay, maybe I want to participate in both of these programs, you can do that if you're going to work on an independent project by simply joining the research internship. And then you can actually participate in many of the sessions that are designed to train you or to guide you in your project preparation process. So again, the research internship link is also available. Let's take a look at that link really quickly on the pre-registration for this summer internship. So again, this is a rolling enrollment. So there's not necessarily a starting date. We kicked off the program in June, but we have people that joined even before that. What you will get here is access to all of our materials, all of our tools. It will be completely online and remote. It is a paid internship, so you have to pay for access to some of these programs and tools. But once you join, again, once you fill out this form, we are happy to continue and send you additional information on how you can get the most out of this opportunity. Okay, so are there any other questions that I can answer at this time related to these upcoming programs or related to the internship program if you're interested in participating in an independent research project. Okay, so I encourage all of you to stay in touch with us. You probably have seen some of our social media posts, and we have a lot of free resources available as well. If you would like to join and check the quality of the materials and make sure that this is the type of course that you will benefit from, I encourage you to sign up. To sign up, let me just briefly walk you through. We have a number of different portals designed around specific audiences. So the first portal that we will use during the programs that I highlighted today is called Science Coach. It's done in collaboration with an organization that deals with students in schools that do high-level research. And here on this site, you can see that I am logged in. So once you create your profile, you can change, obviously you will set your password, you will join a specific course that will start appearing on your profile. So if you register for free, you can already start accessing some of the materials, and that includes Introduction to Bioinformatics and some other introductory courses. As you see some additional courses that you're interested in, you can either purchase them individually, or you can get access to all of these courses in the context of these programs that I highlighted. After you do that, you will start seeing points accumulating on your profile. And you'll also see how these points actually compare to some of the other participants of a program, which is what we use them for, but also help us track all of your activity. And that activity includes the different pipelines that you run, the different codes that you try, and we can then address any of the issues that happen around those pipelines. So really, it's more than just a portal with courses. It is actually a portal designed to build up a community. So this community is specific for high school students interested in research. And we are now just recently expanding this for those types of participants. We have another site that is very similar, and that site is our main portal for all of our online participants. And we have many programs that run from this main portal, which is called edu.t-bio.info. And here you can see some of the participants, especially those that have been highlighted. Here in our blog, we regularly highlight some of the participants of previous programs, and we post all kinds of announcements related to the programs that we have. So I did want to highlight this recent blog post by one of our participants who's a bioinformatician at Atlas, a company that does genetic testing. And you can see from his perspective why he decided to join the OmicsLogic training, as opposed to, as you know, there are many available courses that you can take. And so you can read a little bit about his background and what he has been able to achieve, including how he has used his experience in this program to design a program of his own. And he shares even here some of his notes from some of the sessions. So you can see here how he has been participating in some of these discussions that we had during the session. So you can get a sense for what those sessions cover and how the different assignments work, how we review the different elements. Here specifically, you can see about COVID-19, and then how obviously you can, what type of takeaways you would have from this program. So I encourage all of you to take a look at some of those reasons. Again, I believe that we are putting in a lot of effort into making these programs effective and not just post out there some materials that you can find anywhere online. Okay, so any other questions about these programs? I'd be happy to answer them now. Okay, I'll give it a couple of more minutes. Deepsa, if you have any questions there on YouTube, please share those with me as well. If there are no other questions, thank you for joining today, and I will be looking forward to interacting with you during the program. I will be also personally present in many of those. Okay, Esther is asking, is it PC required or can the courses be managed on an iPad? Yes, all of the coursework can be managed on an iPad. In fact, even some of the materials on the platform you can access on your iPad. I do not think that we've done sufficient testing on an iPad because obviously to practice some of these assignments, you would have to download and upload different data sets. And I know that's a little bit complicated when you have an iPad. So if you want to try, we're happy to maybe work with you and see how it works. But you can definitely try the free course for now on Introduction to Bioinformatics. See how that works on an iPad. I'll show you here briefly. Introduction to Bioinformatics is a course that talks about big data and different sequencing technologies and different types of applications of bioinformatics in different domains. You can see that a lot of the coursework will be videos, and some of the introductory, specifically in the introductory courses, are focused on materials to help you get started, understand the different applications, and practice, evaluating what you have learned. As you go through the more advanced courses, they start giving you assignments, and those are graded assignments, but they will be, related to downloading some data sets, doing some modification to those data sets, then uploading the results and things like that. So that's the issue regarding just having iPad access. Okay, if there are no more questions, I am going to look forward to interacting with you again during the programs if you decide to join. And if not, I still encourage you to take some of the free materials related to bioinformatics and different types of omics data sets. As you go through those, we are going to continue and send you upcoming program information if you are going to be interested in some of the other programs that we will have coming up, either in August or coming up in the fall. We have a lot of new material that is coming out, and we are excited about how that material will help prepare the next generation of bioinformaticians. So again, thank you for joining today. We will be putting the recording from this webinar on our YouTube channel. The YouTube channel also includes a lot of materials that you will find related to bioinformatics and to different aspects of what we do on a regular basis. So obviously, those of you that are on YouTube, you already have the channel. But please take a look at some of those materials there as well. Thank you very much, and have a great rest of your day.